I'm subbing in for Dr. Miller today, so I apologize for not taking anything, but I see you're enjoying your donuts, so that's good. And without any more delays, Rachel is going to tell us all about how we can use gold nanoparticles to make an impact in breast cancer. So, okay, take it away. Thank you.
All right, so good afternoon, everyone. My name is Rachel Tinky, and today I'm going to be talking to you about my senior research project that focused on the synthesis of gold nanoshells for applications in photothermal therapy of MDA MB231 breast cancer cells. So before I begin, just by show of hands, how many of you guys know what a nanoparticle is? Okay, so we got a lot of people in there. So, but if you were in my position junior year, you'd have no idea. I literally had no idea what they were. So as I progressed on my research, obviously I gained more information, and I'm happy to tell you that I can now explain what they are. So nanoparticle is defined by Jenkins and colleagues as something that is relatively small, so it's right in the range of that 10 to 200 nanometers in size. So the question then becomes, how small is small? So if we look here, we can see on this x-axis we have nanometers. Then we have some familiar items. We have a water molecule here, about 10 to the negative first nanometers, glucose, and then as soon as we start reaching the point of antibodies and viruses, we see that right within that range is where our nanoparticles actually fall. So an antibody is roughly 10 nanometers, a virus roughly 100 nanometers. However, we can't physically see an antibody or a virus, so it's difficult to wrap our brains around exactly how small a nanoparticle is. So what we can do is instead compare it to something that we're all very familiar with. So most of us have hair. The majority of us, everyone in this room does. So if we go and take a look at the average strand of a human hair, the diameter is roughly 100 microns. So if we think of a micron, that's 10 to the negative 6th. Nanometer is 10 to the negative 9th. So if we have a 100 nanometer nanoparticle, it's roughly 1,000 times smaller than the average diameter of your human hair, which is really, really small. So along with this sort of diversity in size range, we also have a diversity in the types of nanoparticles that we can have. We could sort of categorize them by inorganic and organic. However, these categories don't necessarily relate to where they could be applied. So we can actually use inorganic nanoparticles within the body as long as the materials are made of are biocompatible. However, there is one such nanoparticle that a lot of people up here in Dr. Miller's lab would be familiar with, and that is a liposome. So liposome is essentially a phospholipid bilayer that is surrounding a water molecule. And I know that Logan and Hannah, you guys are actually trying to load the liposomes with curcumin to get certain therapeutic effects. So this is actually an example of how nanoparticles can be used for drug delivery. However, within my specific research, I focus more on the inorganic side of things, and in particular with gold nanoparticles. So gold nanoparticles can be synthesized in a couple different ways, in a couple different forms, the most common being just a solid gold colloidal nanoparticle, which is what is pictured here. But we can also synthesize them in what is known as a nanoshell form. And this is described by Fukugami and colleagues as basically a nanoparticle that has a dielectric core, and in our case that's going to be silica, and then surrounded by a thin metallic layer, which in our case is a shell structure made out of gold. So then the question becomes, well, why would we want to use the nanoshell over the solid gold nanoparticle? And really, it's because there are a couple different advantages of metallic nanoshells that we can exploit for uses such as in biomedical applications. So Khan and colleagues actually named some of these advantages within their research. The first of which is size control. So silica is actually a lot easier to control the size. So for example, you can just change ratios of reagents. You don't actually have to use something like a size extruder, for example, that you would use with a normal gold colloidal nanoparticle. And that also speaks to its ease and preparedness. So essentially, what we're going to see as I go through the synthesis a little bit later on, a lot of these reactions are self-assembly mechanisms. So basically, we just add reagents into solution, allow them to stir, and we get formation of the product out of that solution. Furthermore, nanoshells are also cost-effective because the bulk of the nanoparticle is actually silica, which is more abundant than gold. It's cheaper than gold. And then we also have to use less gold on the outside to create that shell as opposed to just making a solid gold nanoparticle. So we could save some money in that aspect. Furthermore, gold and silica are also very biocompatible, so we can use them within the body for biomedical applications. And then one of the most important characteristics that we can actually get with these nanoshells, in particular with the gold, is surface plasma resonance. So on the surface of this nanoparticle with the gold, there's an abundance of electrons. And what we can do is actually hit this gold surface with a wavelength of light and get vibration of those electrons that allows for absorption at that wavelength. So typically a normal gold nanoparticle would have a resonance at roughly 550 nanometers, and it's pretty much going to stay consistent throughout the entire solution. But what's really cool with nanoshells is we can actually manipulate this resonance through increasing the shell thickness. So, for example, if this is the shell that's not complete shell, so it might resonate at about 500 nanometers, but as we increase the thickness, we can actually get a shift towards the red infrared region, that's known as a red shift, to longer wavelengths. Typically within this 600 to 800 region is where we want to be. And this is really important for biomedical applications, and in particular with photothermal therapy. So photothermal therapy is described by Riley and colleagues as an introduction of the nanoparticles into the tumor tissue. So this can be direct injection or an intravenous injection, and recognize if you're going to be giving it through the vein, you're probably going to need a specific receptor on the outside of that nanoparticle to actually get to the tumor site. However, what we're hoping for is aggregation of these nanoparticles within those tumor cells. And then following that, we can actually hit it with a laser that has a wavelength of it in that near infrared region and cause vibration of those nanoparticles. And a byproduct of that vibrational energy is heat. So what we do is we induce an increase in temperature within those tumor cells that eventually induces apoptosis and tumor cell death. However, aggregation is very important because we need to be able to create enough energy and have enough of those nanoparticles concentrated in that area to actually increase the temperature enough to get cell death. And primarily the reason we might want to employ an alternative cancer treatment such as this is because it gives a lot more specificity and targeting. So instead of traditional cancer therapies such as chemotherapy or radiation that basically target all cells, we can specifically look at targeting tumor cells, and we can avoid some of these types of side effects such as hair loss, mouth sores, trouble breathing, weakened immune system that are commonly seen with traditional cancer treatments. So with that being said, the overall goal of my research was to synthesize gold silica core nanoshells, hopefully getting full shell coverage with a thick enough shell to get resonance in that near infrared region so that we can apply it to photothermal therapy of our MDA-MB231 breast cancer cells. And the reason that we're using these types of cancer cells is because they're readily available within the lab, and it's what we pretty much have to use. So as we move forward, within the synthesis of nanoparticles in general, there's two basic approaches. The first is a top-down approach in which you start from a bulk material and essentially break it down into its individual components through instrumentation, eventually reaching that atomic layer, and then we can take that atomic layer and build it back up into our nanoparticles. However, it takes a lot of time, and it's also pretty costly because you have to start with a large material, you have to purchase a large material, and you also have to have the correct instrumentation to actually ionize it down to get those powders and eventually get those atoms. So instead, what we're going to employ is exactly the opposite of this. So we're going to start from the bottom up, essentially. So we'll start with those very small molecules and be able to build our nanoparticles from there. And as I said, it takes a lot less manpower. You don't have to have all that instrumentation. And you can also start with smaller amounts of reagents, so it's very cost effective as well. So within the synthesis of gold nanoshells in particular, we can actually break this down into 4 distinct steps. The first of which is synthesis of the silica core. We then want to actually modify the surface of that silica with an organic group. And this organic group is going to be in the form of an amine. And the reason we want to use amines is because amine groups have an affinity for binding gold, but also when we initially introduce the gold onto the surface, it's in the form of chloroauric acid, which is negatively charged because the gold is surrounded by chlorine molecules, as we'll see a little bit later. So the amine is also going to give a positive charge in solution, which can readily bind and create an even larger affinity for that negatively charged gold particle. And then finally, after we seed it with just a small amount of gold, we can build from there and get reduction of larger layers of gold to build up that entire nanoshell. So, starting out, we're going to use silica core synthesis, and to do this we use tetraethyl orthosilicate. It's a mouthful, but basically this is just our silica that we're going to be adding into solution. And what we can do is mix that with specific ratios of alcohol, ammonia, and water, allow that to mix in solution, and eventually we will end up with a solution that looks like this, that's white. And this method is actually a modified version of the Stover method that was initially introduced by English and colleagues within their synthetic process. So this white coloration is what we're looking for. We can actually see solid particulate in that solution, which is indicative of that silica core formation. Following this, what we're hoping to see is eventually we have these hydroxyl groups on the outside of the silica so that we can eventually modify the surface with our organic layer. So to do this, as I mentioned earlier, we want to be adding amine groups on the outside, so we use 3-aminopropyl triethoxysilane, and the binding is going to occur basically in this area in which we'll have a free amine coming off of the surface of that silica particle. And eventually what we hope to see is our nanoparticle with an abundance of these organic layers coming off. So to do this, we start with just our silica nanoparticle. Then we introduce our APTES in excess because we want to make sure that we have enough bound to the surface so that we can eventually bind gold in the following step. And then we actually have to add heat. And this heat is important because it allows for that covalent bond formation to occur. Following this, we then can purify this solution because we want to make sure that we don't have any excess APTS in solution. So we can actually centrifuge it, and this was a little bit time consuming. This was 9 centrifugations of 10 minutes, and then redispersing within water. So that was fun. But eventually what we're left with is this nice pellet which we can redisperse in solution to use later on. So the question then becomes, how do we know that we're making what we think we're making? And we can actually use a couple techniques to characterize these particles, the first of which is actually our UV spectroscopy. And the way that this works is we have a light source that you can pass through a monochromator, and a monochromator is essentially like a filter, so you can select for the different wavelengths of light that you might want to look for. That's going to pass through and hit the sample, and in some cases you have a reference cell, which basically takes away that background noise. So any signal that you'd be getting is actually from your sample. That will then reach a detector, which eventually will give us data output in a computer. However, I was actually lucky enough to use a biotech plate reader, so I didn't have to use those cuvettes. Basically, I just loaded all my samples into a 96 well plate, stuck it into the plate reader, was able to select my different wavelengths that I wanted to look for, which was in the range of that 400 to 900, because gold, as I mentioned before, resonates around 500, but we're looking for a little bit more of that redshift towards that 700 range. So eventually, I was able to get absorbances within those wavelengths. So while UV-Vis is very good for telling us if we have gold in solution, it doesn't necessarily tell us if it's actually bound to our silica. So instead, we use a complementary techniques of scanning electron microscopy as well as transmission electron microscopy. So these diagrams are a little bit complex. However, the important information to know is that scanning electron microscopy uses the scattering of electrons. So eventually, we're going to get this kind of focused beam that's going to hit our sample, and it's going to tell us a lot about the surface characteristics, so we can get 3D images from there. However, we can't really see a lot within our silica, or within the structure that we're making, so we use transmission electron microscopy for that, in which it actually uses the transmitting of electrons through our sample, and it basically gives us an image that looks like a slice. So if I were to cut the silica in half, we can see sort of the outer layers, but we can also see what's going on inside. So for example, this would be a common image that we might see of a silica nanoparticle from our scanning electron microscopy, and this is sort of what a transmission electron microscopy image would look like of those silica nanoparticles. So we do actually have an SEM here at the University of Mount Union. However, as the resolution was very poor. So when we were getting these types of images, we sent them out to Miami University to have this imaging done. But to begin, if we look at this UV vis of our silica core, we would expect to see no peaks within this line. And that's because silica is an organic substance, and so it's not going to have any resonance within that UV range, excuse me. So this is actually very good that we don't see any peaks, and we can confirm that we've actually made the silica through our SEM and TEM imaging, and these are actually my nanoparticles that we sent out. So what we're looking for is very monodisperse. By monodisperse, I mean we want them to be the same size and same shape within solution, and we can see that that is the case for the most part. I mean, we do have some that are a little bit smaller or larger than others. However, they look very nice in the TEM imaging, which is really cool to see. So moving forward from this, now that we have confirmed that we've actually got our SiO2 nanoparticles with those amine groups on them, we can then go ahead and start the seeding process. And this is done through, essentially what we start with is just deionized water in solution. And then we add a little bit of sodium hydroxide along with THPC. And this is a very strong reducing agent to get that gold to actually bind initially to the surface. So from here, we have all that in solution. We can then add in our chloroauric acid. As I mentioned earlier, this is negatively charged, so that's why those amine groups are important. It's going to allow for that initial binding to those positively charged groups in solution. And then we can go ahead and add in our silica modified nanoparticles, allow that to stir around in solution for a period of time, and then eventually we get the self-assembly of those small amount of gold particles binding to the surface. So we get a solution that looks like this. It's very dark brown. It started out clear, and then it turns this color. And unfortunately, we have to purify this one again, so I went through another round of 9 centrifugations of 10 minutes. And eventually ended up with a pellet that looked like this, which I redispersed in solution to get a UV vis of. And if we look here, we would expect to see maybe a small peak of that gold. So the orange line is corresponding to that silica core. This gold seed solution is indicative of this blue line. And we maybe see a small peak here, but because we're only binding very small particles, it's likely that the gold is actually below the limit of detection for this instrument. So to actually visualize the gold binding to the surface, we have to use SEM imaging. So if we look here, this large molecule is our silica. If you look very closely, you can see little white specks that are actually bound to the surface, and that's our gold. So that's how we can confirm that we actually have gold bound, and we can continue moving forward with our synthetic process. So just a quick recap, we have our gold, or we have our silica with our metallic seeding, and now we actually want to start building that shell layer through the reduction of those metal ions onto the surface. So to do so, we introduce the gold through a potassium gold solution into our seeded nanoparticles, and eventually reduce that gold with ascorbic acid onto the surface. So this was done for a period of 2 trials, so I went through the process twice. And the reason that we want to go through the process twice and we can't just reduce it all onto the surface to begin with is because if we put that high of a concentration of gold into solution, it's not going to want to form a shell because it's going to be interacting with itself. So what we'll actually get is just solid gold nanoparticles instead of shell formation. So this was the resulting solution of my 2 ascorbic acid reductions. And this purplish pink color is actually indicative of shell formation. Now the pink is a little suspicious because normal gold colloidal particles are red. So this leads me to believe that there might be a little bit of unreacted gold in solution. And if we look at the UV vis, we can actually see that that's true. So what I actually did was just centrifuge at one time, take a sample from the supernatant, and then take a sample from that pellet. Our pellet should be the nanoparticles themselves, and there shouldn't be any nanoparticles within our supernatant. However, what we can see is we do have a small peak right around that 500 range, which does indicate that there is some unbound gold in that solution. However, there is a much larger peak in our pellet, so that leads us to believe that we actually did have gold binding to that surface, which is good. So if we go and look at the SEM, we can confirm that we actually have gold bound. So all this white is gold that is bound to our silica nanoparticle, which is really, really cool. And don't know about you guys, I'm a nerd, so so cool to see this, but we can still see some gaps. So we don't necessarily have full shell coverage, which is what we want if we want to move on to our biomedical application. So instead, what I actually did was do a third ascorbic acid reduction, in which I got this nice dark purplish blue color, which I was really excited about. So we sent this off for imaging as well. And I did the same thing that I did with the two ascorbic acid reductions. I centrifuged it one time, took a sample from the supernatant pellet, and what we can see is that this blue line is a pellet. So we have a nice full peak right around the 600 nanometer wavelength, which is important, and we're going to touch on that just a little bit later. However, we can see that in the supernatant we don't have any type of peak, which is really good. So we can see from the UV vis or infer that most of that gold has bound to the surface of our nanoparticle. And looking at our SEM, we can see that it's pretty much all white. So we did get full shell formation, which is really, really important when we move on to our photothermal therapy techniques. However, there was one more thing I want to point out. If we actually overlay the UV vis from our second ascorbic acid reduction and our third ascorbic acid reduction, we can actually see this red shift. So the 2 times reduced is right here in blue. It's resonating at about 550 nanometers. However, our third is actually resonating at about 650. So that's why it's really important that we get that full shell coverage, because now we're moving into that near infrared region. So we can actually use those nanoparticles for photothermal therapeutic treatments. So just a quick recap. Photothermal therapy works through introducing those nanoparticles into the tumor tissue. We can then hit that with a laser, and we want to have aggregation so that we can get enough energy to increase the temperature to eventually result in tumor cell death. However, there is a slight issue with the way that we actually introduce the nanoparticles into the tumor tissues. So, but the human body is primarily made of water. So when we actually introduce the nanoparticles into the tissue, we dilute them. So instead of having that concentration of aggregates, the nanoparticles can actually spread out. And then we don't get as many nanoparticles getting into those tumor cells as we would like. So that causes not enough energy to be formed to actually get enough increase in temperature to kill off those tumor cells. So what we want to do is find a way to promote aggregation. And basically what we can do is actually create an aggregation template, and this was first introduced by Wong and colleagues, in which they were trying to figure out how to increase the efficacy of photothermal therapy. And basically there are a few characteristics of an aggregation template that we want. We want to be able to bind nanoparticles, first of all. Second of all, we want to have multiple binding sites so that we can get groupings of those nanoparticles so we can promote aggregation. And thirdly, we want it to be biocompatible, because we're going to be putting it in the body. We don't want to have any cytotoxic effects to surrounding healthy cells. So what they actually did, what the research actually did in the Wong article, was they came up with using a silk protein that was fibrin. So what they actually noticed was on the surface of this protein there was a lot of acidic residues in the form of glutamic acid and aspartic acid, which is negatively charged in solution. So what they decided to do was actually modify the surface of their gold nanoparticles with polyethylenamine, which gives us a lot of these amine groups, to make the nanoparticles positively charged so we can get electrostatic binding to that fibrin protein. Not only does it give a positive charge in solution, but we know from previously that amines have a high affinity for binding to gold anyway. So what we're hoping to see is through this modification, what we get is this gold nanoparticle with a lot of these polymer pieces coming off of it that are going to cause it to be very positively charged in solution. And what the hope is that if we introduce this negatively charged silk fiber protein with these positively charged nanoparticles, we can actually promote binding and grouping and aggregation of these nanoparticles, in which then we can introduce this entire complex as a photothermal treatment. And the reason that this will work as well is because tumor cells, while stuff can diffuse in and out the nanoparticles, this is a little bit larger, but tumor cells readily uptake stuff from their cellular environment. So we can get intake into the tumor cells of a little bit of a larger complex because of that. So these were actually their results that they got from their article. And this is a circular dichroism graph up here in the left-hand corner. And circular dichroism is just another type of atomic spectroscopy in which you can actually get a little bit of information regarding binding. So if we take a look at this red line, this is just the fibrin protein by itself. However, upon binding to that gold, we do get a shift in wavelength. So that's what they used to sort of see if gold was binding initially. And then to confirm that, they used just light microscopic imaging. So if we take a look at B, this is just the gold nanoparticle in solution by itself. And what we can see is that it's pretty dispersed. There's not a lot of aggregates forming. However, when we introduce the silfibrin protein, what we get is binding to that surface, and we see a lot of groupings of these nanoparticles together. So panel C was a 50 millimolar concentration of that fibrin protein. D was a 100 millimolar, C was a 150 millimolar, and F was a 250 millimolar solution. So right within this 50 to 100 millimolar solution, we see significant aggregates being formed. However, if we get too high of a concentration of those long silk proteins in solution, basically nanoparticles still bind, but they're so spread out that we don't get aggregation anymore. So we know that, so they found from their research that the optimal concentration for this type of aggregation template would be roughly within that 50 to 150 millimolar range. So what Dr. Miller was initially proposing for my research project, when we were talking about application, was using extracellular DNA for macrophage activation. But after reading this article, what we realized was that DNA is actually very similar to this fibrin protein. You can get very long strands of DNA, for the chromatin DNA, and it also just so happens that DNA is also negatively charged due to this phosphate sugar backbone with this phosphate group. So instead, we sort of changed our idea into the proposed use of DNA as an aggregation template with my nanoparticles to try to get photothermal therapy of breast cancer cells. So this was our goal. What we would hope to see eventually is that we have our DNA and then we have this gold nanoparticle with this positively charged surface bound to that DNA. And this figure is a little bit misleading because this is a relatively short segment of DNA. We would hope to get very long strands so we can get multiple nanoparticles actually bound to get those aggregate properties. So moving forward, to actually carry this out, I went ahead and did some genomic DNA isolation. And I used E. coli to begin with because we have a lot of that available. So if I messed up, we wouldn't be wasting any of our cancer cells, which is hopefully the direction we want to move toward, is actually using mammalian DNA for this process. However, we first started with E. coli, in which I used, I lysed the cells open, removed protein with actually phenol and chloroform, and then went ahead and precipitated it out with ethyl alcohol and rehydrated it in a TE buffer so that I had a nice solution of DNA. And I ended up getting it roughly around 210 micrograms per milliliter, which is fairly concentrated. And then moving forward, I actually had to modify the surface of my gold nanoparticles to make them positively charged. And we decided to use the same polymer that was used by Wang and colleagues of this polyethylamine. We bought multiple molecular weights of it. However, I started out with the shortest, which was around 600 molecular weight. And hopefully what we're going to get is this gold nanoshell that again has those polymer groups coming off of it. So in order to facilitate this, hopefully with our aggregation template, I went ahead and used a 96 well plate, and I introduced different concentrations of DNA. So I started out with a non-dilute concentration and then did a subsequent 100-fold, and from there did serial dilutions of 10-fold from there. So I ended up with about 5 samples. And to that I added consistent volumes and concentrations of those gold nanoparticles with the PEI modification. So from there, what we're able to do was actually take inverted microscopic images. So this is an image we were able to get from our gold nanoparticle control. As you can see, there's still some aggregate, a little bit, within the solution itself. So this is what we would be comparing to. However, most of those particles are very, very small. And this was our undiluted DNA control. And we saw some of these like white particles in here that were fairly large. And we're not sure if that was actually aggregated DNA or if it was some type of contamination possibly. because we don't necessarily see this within our undiluted samples, but it could just be that the concentration is too low for that DNA aggregation to occur. So when we actually look at our lowest concentrations of our DNA and gold nanoparticle, they looked very similar to the control, in which we saw just about the same amount of aggregation. It wasn't until we get to our 1,000-fold DNA dilution that we start to see these larger groups of aggregates being formed, in which we see longer chains compared to our gold nanoparticle control. And what's interesting to note is that this is actually the opposite of what we would expect from the previous research article, in which they actually saw higher aggregation at lower concentrations. But because we use the coli DNA, those segments are so short, they're about 1 micron long, that we would actually probably need a lot more of that DNA in solution to get aggregates to form. And the reason we're seeing sort of these short changes is because of that small length. But as we move forward into our 100-fold dilution, we can actually see that there's a lot more of these larger groups being formed in comparison with our AU nanoparticle control, which was cool to see. So this is basically where we are right now with this project. There's a lot of preliminary data. So we do have a lot of future directions that we would like to go. The first of which is increasing the concentration of DNA that we actually initially extract, because when I did the extraction method, I diluted in about 6 mils. We're hoping we can dilute it in even smaller volumes so that we get a higher concentration from the very start. Following there, we can also modify with different PEI lengths. As I mentioned before, we have those longer chains that we can try to see if we can optimize aggregation as far as with the surface modification of those gold nanoparticles. We can also assess the effect of pH on the aggregation. Because we are using electrostatic interactions, pH may play a role in that. And then we can ultimately, what we want to do is be able to utilize an NIR laser to assess the ability to actually kill those cancer cells. In our physics department, we actually have a 780 nanometer laser that Dr. Teeling-Smith was gracious enough to help us set up a little bit earlier this week. So it was a little bit dim in there, so it's hard to see. So I'm going to point out the components. This is our laser, and then this is our infrared gun, which we would try to go ahead and measure heat with. But basically, we'll take this laser, hit it off of this mirror, and basically get it to go straight down so that we can have our 96 well plate sitting underneath it, and we can concentrate that laser into each well for a select period of time to assess the ability of the nanoparticles to create a change in temperature and eventually kill those cells. So moving forward, what we also would like to look at is within our synthetic process, if we can actually make nanoparticles with different metallic shells. And I actually had the opportunity to work a little bit with palladium shell growth. And the reason we want to use palladium is because there's not a lot of research out there regarding the ability to get nanoshells made of palladium. And palladium can also actually be used within electrochemistry as a catalyst. So to do this, what I did was just had a palladium chloride solution. I actually combined it with the gold seed. I tried to do a palladium seed, however, it was a complete failure. So I decided to go ahead and try to use the gold seed and then reduce it with ascorbic acid. And then what we ended up with a solution that looks like this, that was black. And when I actually centrifuged it down, I got this nice black solid, which was indicative of palladium shell formation. We sent this one out to Miami as well, and we actually did get full coverage on the very first try. But there is a lot of unreacted palladium within that image, so that probably speaks to the fact that the chloride solution that I made was probably too concentrated with that metal. So this could be a future project that students could work on with respect to electrochemistry, as I mentioned before, and actually using it as a catalyst for those types of techniques. So with that being said, I would like to thank the University of Mount Union Chemistry and Biochemistry departments, all of you guys for taking the time to listen to my spiel here. I really appreciate it. I'd also like to thank Dr. Jean and Dr. Miller for all their help. Dr. Jean has been with me from the very beginning since my junior year. So thank you so much for all your guidance. Everyone in the Miller lab that helped with the extraction of DNA. Jay, who is my co-captain on this project moving forward. Thank you so much for all your help. And I'd also like to thank the physics department and Dr. Teeling-Smith for helping us with the laser setup and utilization of that. These are my references, and with that, I'll open up to any questions. Yes, bro.
So with the silica particles, you got really good monodispersity.
Right.
Were you worried about monodispersity putting the gold as a shell, or was it going to be monodispersed because of the silica gel particle?
Yeah, basically the metallic layer is so thin that it's not going to really affect the aggregation as much with those silica particles. Yes.
So for your NIR laser, is that what it's called?
Yeah.
Do you know if it would have any negative effect on your nanoparticles in solution? I know you're wanting to kill your cancer cells with it, but would they degrade, like photodegrade after so long being hit with a laser, or have you read any side effects about that?
Actually, the reason we want to use NIR, I don't know if I mentioned this, is because we can get penetration into the tissues. So we want to have a prolonged exposure to that NIR to make sure we get enough energy in that resonance in that gold. However, I didn't read anything as far as like degrading of that gold, but they use, I think, an exposure time of around 6 minutes to get enough energy to raise the temperature at those things. Yeah, it was a long time solution.
So whenever you went to make your palladium nanoparticles, did you also do like
three reductions?
Yes. There you go.
No. So if I go back to that, this was sort of what I was talking about before. We didn't get shell formation. The solution I used was so concentrated that I was actually able to get full shell formation. But I also got a lot of these colloidal palladium nanoshells in solution, which is what I didn't want to happen. That was unbound. So we probably want to lower that concentration and do multiple reductions. So we're just left with this palladium nanoshell structure instead. Right. Yeah. So that was one of the issues. That's primarily why we had to do the multiple reductions with the gold, is so we didn't have this happen.
Do you have any idea how big your gold seed particles are? Like what those are supposed to?
You say 1 to 2?
It's really tiny.
You're really small.
Okay.
Yeah.
Up to 5.
Because the entire shell, or the entire silica is
right.
roughly like 200 nanometers. So yeah, they're really small. Yeah.
Was it expensive to use the gold?
Not that. I don't know. Dr. Gina was in charge of getting them, but we only needed 1 gram of it to be able to get our actual solution, so it was relatively inexpensive. And the silica, they said, is pretty abundant, so we didn't have to spend a lot of money there. Yeah. Any other questions? Go ahead. Hannah.
I know that when you're using nanoparticles and that sort of thing for drug delivery, you care about them staying relatively small. Is that a factor in your research at all?
Really, the size is not a particular concern because we want aggregation, so we kind of want them to clump together a little bit. That's primarily the reason that we want to use that template. They're still going to be small enough to where they're not going to really cause any adverse effects, but we do want them to be a little bit larger to get that clumping within the tumor cells themselves, because we want enough energy to actually change the temperature. Yes.
Has this research been done with any other like metal ions, or has it? Why did you choose gold?
Gold, there's actually a lot of research out on there, and it's very biocompatible within the body. That's one of the reasons that we actually want to look at the synthetic technique with other metals, because there's not a ton of research out there as far as, can different metals be used for biomedical applications, such as like palladium. But I know Dr. Jean wants to look at palladium as a catalyst as well, so there are a lot of different applications of different metals, and that's sort of why that would be a future project moving forward. But yeah, gold is just very biocompatible, and there's a pretty nice synthetic process to be used for that. Dr. Mason, did you have a question?
Well, one of them was kind of related to
Oh, okay.
gold, but I have one way back.
Okay.
What's the interaction that you start with silica and add the APTS? Why is the APTS? What's the interaction between base silica and then APTS? That's way back.
You get a covalent bond essentially being able to be formed. this is sort of what you're looking for when you have the interaction between the silica and those hydroxyl groups. So you get essentially covalent bond formation through the addition of heat, which is basically what we're looking for. So if you remember, like, this is the structure of APTES. See this silica here? But then on the surface of the silica, you actually have all those hydroxyl groups. So you get binding of that silica to those oxygen groups on the silica surface. And you have to add heat to get that covalent interaction to actually occur.